Hello, everyone. Welcome to tonight's Save Our Seas Distinguished Speaker Series, July edition. I'm Lance. I'm from the Museum of Discovery and Science in Fort Lauderdale, Florida. And tonight we are joined by Dr. Cheston Peterson, he will be giving you a lot of really cool information about some of the work that he's doing with bonnethead sharks in Florida, ecology, estuarine ecosystems. Really fascinating research. I'm really looking forward to hearing tonight. So welcome for everyone that's here. A few little things about tonight. Please feel free to ask questions. We want this to be interactive. So if you have a specific question you'd like to ask, please type it in the chat. We will be doing questions later on as well, but as well during the presentation. If you have a burning question you really want answered as Dr. Peterson is presenting, please put it in there. We'll see if we can get to it as we're going along. So don't feel like you have to wait to the end. Please ask questions if you have them. I'm going to ask that everyone keeps their microphones on mute. You're more than welcome to turn your cameras on if you would like. It's always nice to see your smiling faces, but you don't necessarily have to, of course. A few things about the museum and this program. We are very honored to be sponsored by the Save Our Seas Foundation in doing these talks. The museum itself is, like I said, based in Fort Lauderdale. The mission of our museum is to connect people to inspiring science. And we do that through 4 different pillars here in the museum that kind of drive everything that we do. Our strategic action pillars are physical science education, early childhood education, health and wellness education. And where these talks fit in is into our environmental sustainability efforts that we are doing here at the museum. I am the director of environmental sustainability here at the museum, and we're doing all kinds of things from paid internship for high school students to bringing in world-renowned scientists to spread information all about ecology and conservation of the natural world. So we're always looking to do fun things and educate the public wherever we can. So with that, I will introduce Dr. Peterson, and we'll turn it over to him. So we'll go, Cheston is a marine fish ecologist who specializes in coastal sharks. He's been fascinated by the natural world from a very young age, but nothing has captivated Cheston like water and everything that lives within it. When he was growing up in Tennessee, his first loves were frogs, and especially salamanders, but later transitioned to fish, sharks, and rays became his favorite things to look into. During his undergraduate studies, his interests became more focused on fisheries management and conservation. Doing it through volunteer work, Cheston gained some positions through the Moat Marine Lab, and an internship with them, and eventually found himself on his way to graduate school. Cheston's been involved with several projects during his master's and PhD, most of which involve or revolve around various aspects of coastal shark ecology, from topics such as spatial patterns of distribution, where the sharks are, and abundance of fishes in Florida, especially in the Big Bend Panhandle area. Cheston has tried to find the intersection of the basics of applied science and maintain a balance that work by pursuing questions fundamental to ecology while collecting data that are also useful to management and conservation. So marrying those two things of useful science, right, where we can take it out into the field and use it for something to make our world better. So that's really awesome. I'm really excited about that. So without further ado, Cheston, welcome to the Distinguished Speaker Series.
Thank you, Lance. I'm excited to be here. I appreciate that introduction. That was great. I'm going to share my screen here.
Yeah, go ahead, share your screen. And like I said, if anyone has any questions, please feel free to put it in the chat and we will go through, all right?
All right, so thank
There
you
we go.
again,
We can
Lance, for that
see it.
introduction. Thanks everybody for coming. I appreciate you taking time to tune in and hear me speak. I'm really excited to be invited to do this and give this talk. I think this is a great series and just excited to be a part of it. So as Lance said, I'm going to talk to you guys mostly about bonnethead sharks in Apalachicola Bay. The title of my project, which was sponsored by Save Our Seas, was Eat or Be Eaten, Effects of Prey and Predator Distribution on Movement and Habitat Use of bonnethead sharks. And as Lance said, feel free to throw a question at me. And if there's any kind of terms or jargon I'm throwing around, just to clarify, just speak up. And so I'm going to give just a brief introduction about why we study movement and how we study movement of coastal sharks, which can be studied through a variety of means and scales. Conventional tag recapture and long-term monitoring are what you might see a lot of, where we just simply put a tag in a shark, and if it's recaptured by fishermen, by other scientists, they hopefully will call that in and tell us where they caught it, how much it's grown, and that kind of thing. So we can get an idea of broad-scale movement, some ideas of seasonal habitat use, things of that nature. Whereas we can use more advanced tagging technologies, such as acoustic telemetry, to look at fine-scale movement, and we can look at diel or patterns throughout the day, or temporal scale patterns throughout the seasons or years of habitat use. And so this gives us much more fine-scale, detailed information, usually over a shorter period of time, where we don't have to fill in those gaps quite as much. And so that's what I'll be talking about and using today. And so I should have mentioned there are a couple of pictures here of just some sharks eaten by other sharks, so hopefully that doesn't bother anyone. But this is a major thrust of this project. So predation risk is an influential factor in behavioral decision-making, and animal movement and heterogeneous or mixed habitats is a really classic topic in ecology. And so a lot of the research done in these fields has been historically with terrestrial organisms such as insects and spiders, looking at how they move through a patchy landscape of, it could be bushes or forest or whatever. And now we're trying to kind of turn those concepts to other species, which may be a little more difficult to study, such as marine fish, and especially sharks. And so we know that predation risk is an influential factor in decision-making, and experimentally predator presence has been shown to reduce time spent by prey in optimal foraging habitat. So this is some very classic work in ecology done with sunfish, a freshwater fish species, where they basically put them in tanks and monitor their behavior in the presence or absence of a predator. And it showed that they essentially reduced how much time they were in the best habitat when the predator was around. And so we also think that animals under an intermediate risk of predation may be more likely to invest in anti-predator strategies. And this is called the intermediate predation risk hypothesis. And this is another kind of crux of my project. The idea here is that if I am a fish, type of species that experiences a lot of predation, usually that is kind of ingrained in my life history. So I may grow fast, mature quickly, and make a ton of babies. And if I do that, the risk of predation individually isn't that big of a deal because there's so many of us and we're so productive. So I, as an individual, may not try to avoid predators and invest my energy in that. Similarly, if I'm so large that the risk of predation is very low to me and I don't have to worry about predators very much, I might not invest in anti-predator strategies for that reason either, right? So the idea is that if you are at an intermediate risk or of intermediate size, you may be the most likely to invest in behavioral patterns to avoid being eaten. So that's really important as I move on to my study species.
Justin, can I ask
so
one
the roles
question?
Of prey and oh, sorry.
Sorry, can I ask a question? Sorry.
Yeah, sure, Lance.
When you're talking about anti-predator strategies, what are some examples of that?
That could be modifying your movement patterns or habitat use is obviously what comes to mind when I'm talking about this, where you're, say, I'm not going to go in the most risky habitat, even if my opportunities to find something to eat are best there. So I'll go somewhere where I'm less likely to find food, but I'm also less likely to be eaten. And so that can be a fine scale thing where it may be how animals respond to the presence of a predator, or it may be a broad scale, kind of like an evolutionary adaptation to avoid high risk areas.
Right. So the animals themselves, in this case, bonnetheads, which we'll get to in a second, I'm sure, but they can remember, essentially, if they've had a risky situation happen to them, and they would not go to that area again, maybe?
That's an excellent question. I don't know if it's remembered, or I think the idea that this would be kind of ingrained. And so
that's interesting.
yeah, so I'm not sure. That's a whole nother level of behavioral ecology to think, why are they doing these things? Actually, I can't answer. So, I guess there could be some triggers for that, because it's always a stress response, right? And so there's usually, there should be a kind of a hormonal cascade that's going on, just like you or me. If we get nervous or something stressful happens, adrenaline starts pumping and our body kind of engages with that reaction. And so there could be things like for pheromone cues or smell cues. If they smell a predator, they're more likely to react. So it may not just be memory. It could be stress, that kind of thing. So but that's a great question.
That's interesting. So, okay. So we're trying to figure out where they go based on their risk assessment, whatever that means to them, right? Okay.
Yeah.
Cool.
Exactly.
I understand.
And so these dynamics in sharks are really poorly understood. The role of predator and prey distribution driving habitat use and movement is not well known. We've seen more of this research, some really more classic or things that have broken into the mainstream would be like the reintroduction of wolves in Yellowstone is thought to modify the behavior of deer, which has led to the return kind of cascading effects of reducing pressures, essentially reducing pressures on plants so that native plants can come back after the deer have kind of hammered them. There's also a lot of research in Africa, in the Serengeti, looking at how, where you have a diverse community of predators kind of affecting each other, and we have these guilds of dominance and that kind of thing. But in sharks, this is poorly understood, and we're just kind of cracking into this. And so now I'll get to our study species, the star of the show, the bonnetheads. So this is one of the smallest, if not the smallest, in the hammerhead family, the smyrnids. They're often mistaken as a baby hammerhead. I get that a lot, but they're the only hammerhead with that the distinct rounded front end on their cephalofoil, or their head, so it gives them their bonnethead name. Primarily found throughout the Gulf of Mexico and the Northwest Atlantic. Typically, they're considered a species of least concern in terms of conservation concern, but certain populations may be doing poorly. This is coming from recent stock assessments. They're caught a lot as bycatch in the shrimp fishery and the trawl fisheries, and so that's a threat to them. And then they're also a blue crab specialist, and so they love to eat blue crabs. That's ubiquitous across their range, and very, very common. And so that's another really important aspect of them. Bonnetheads are a small coastal shark. They're managed in that complex. They only get about 3 to 3.5 feet long, about a meter long, and just really, really cool animals. And so our study system was Apalachicola Bay, which is a large river-dominated estuary enclosed by 3 barrier islands. There's 4 inlets into Apalachicola Bay, but it's otherwise a very enclosed system. And it's very diverse. So habitats include oyster reefs, soft bottom, mud, basically, and seagrass. And so if you're not familiar with Apalachicola Bay, as Lance mentioned, it's in the Panhandle. So this is just an image I grabbed off the internet. But this was a major oyster production area. There's a huge oyster fishery here. And so you may be familiar with the region. This is a highly productive ecosystem, as most river-dominated estuaries are. It's a national estuarine research reserve, and so historically it's very well studied. The invertebrate communities and the small bony fish communities are really well studied throughout the bay. There's been some work done on the sharks and rays, the elasmobranchs, but not as much. There's been more work on the sharks on the outside of the bay, and in St. Joe Bay and some of the neighboring areas, mostly done by NOAA. And so, as I mentioned, there are commercial shrimp and oyster fisheries. There's also a lot of recreational fishing, mostly for sport fish and kind of inshore species like red drum and spotted sea trout. But the whole system is dominated by gaff topsail catfish, which you see my former advisor, Dean Grubbs, there holding a gaff topsail catfish. And so these guys are super abundant. They're actually another part of my project that I won't talk about today, but they're definitely the dominant fish species in Apalachicola. All right, so kind of getting into the thick of the study system here, I had 3 focal species that I looked at their interactions, or tried to look at their interactions. So I've got these 4-letter codes here that are just the first letter of the genus and the first 3 of the species. I use those throughout the talk just to refer to the different species. So bull sharks are C-Lu, C-L-E-U, Carcharinus lucas, and then bonnetheads are STIB, S-T-I-B, Spirina tibero, and finally blue crabs are C-Sap, Calanectes sapidus. And so I have just a really simple food chain here where that blue arrow indicates a direct effect or essentially consumption. So that means, we're looking at a direct interaction there. So Portuna crabs, like blue crabs, make up greater than 70% of bonnethead diet by weight, more than 85% by the index of relative importance by percent, or percent IRI. So there's a few different ways to quantify the importance of prey items based on gut contents. These were some older studies from the 90s and early 2000s. But the point I'm driving here is that they like to eat blue crabs. They like to eat crabs. That's a big deal. And then this is another eaten shark image, but we know that bonnetheads get eaten by bull sharks because we've seen it happen in the field. So sometimes when they're caught in the net or on the long line, a larger shark can come up and depredate them. So we know this is a thing as well. We also know that elasmobranchs make up at least part of the bull shark's diet as they grow. And so all that being said, I'm actually not so interested in how often bull sharks eat bonnetheads or how many blue crabs bonnetheads are eating. I'm interested in indirect effect, or what we call non-consumptive effects. And so this is when the presence or the threat of a presence of a predator may affect the behavior of our focal species. So this is exactly like what I was introducing and what Lance and I talked about. So we're more interested in how bull sharks indirectly affect bonnetheads, which can in turn indirectly affect the blue crabs, and we call that a cascading effect. And so this is kind of the main question here. How do prey density and predation risk affect intermediate predator habitat use and movement? And the intermediate predator here is the bonnethead.
Cheston, can I ask another question?
Yeah, sure, Lance.
So we know bonnetheads like to eat crabs. How many crabs can a bonnethead go through in a day? What would be their?
Oh man,
do you have an idea on that?
I don't know if there have been any like daily consumptive rates. I will say that unfortunately bonnetheads have a pretty high mortality rate, so we do a lot of dissections to collect aging structures and guts to support management. And I will say that every bonnethead I have examined had crab parts in them. Okay. Usually there are more than one. The tricky thing is the carapace of crabs doesn't digest all that quickly, and that will stick around for a while. So right. It's tough to say how long they've been in there, but my idea is that they're never empty. It seems like they've always got crabs in there. So I imagine, it would depend on the size of the shark and all, and the size of the crabs they're eating. But if I had to guess, I'd say, 4 or 5 crabs in a day is a good haul. That may be right. A lot. I don't know. And my guess is that they wouldn't have to eat, like a lot of predators like this don't always eat every day, or they eat in bunches. And maybe you had a good day and I got 4 crabs, and I'll take it easy tomorrow, or I'll be good to go in case that the crab foraging is light,
Yeah. Well, they're pretty active sharks, right? So that they need that ram ventilation. They have to swim to breathe.
Yeah.
Right.
They do. They are ram ventilators. Yeah.
Big energy consumption with that. So they would need to
definitely
eat a lot, right? We had
Yeah,
a comment from a guest saying that it's quite an expensive diet the bonnetheads have, right?
yeah.
um, are
That's true.
bonnetheads the smallest of the hammerhead family?
They are. I believe they are. There's been a couple. There's the winghead shark I'm thinking of. I believe bonnetheads are the smallest. There's just a
Right.
couple like really obscure hammerheads that I forget how they stack up size wise, but I think bonnetheads are the smallest hammerhead.
Cool. All right. Thank you very much. That's another
Yeah.
question from the audience. All right. I'll mute myself.
Okay. Thanks, Lance. So I have a few sub questions to that main question, and that this is kind of all leading up to how can we answer this broader question? We have to answer these smaller ones first. So one is, our densities of blue crabs and the probability of encountering a bull shark heterogeneous? So what I mean by that is, in Apalachicola Bay, are blue crabs just evenly distributed? Is it just completely even? There's as many blue crabs in the north as there is the east or the west, and same thing with bull sharks, or not? And then does the density of appropriately sized blue crabs predict bonnethead habitat use? And that's an important one, because we know that bonnetheads only eat kind of a mid-range of crabs, and there's especially a too big of a crab. So I corrected that in the data. And then, is there evidence that predation risk may affect habitat use in bonnetheads? And how are environmental factors such as salinity relevant? So we have to keep this in mind. In a river-dominated estuary like this, there are some really intense salinity fluctuations throughout the day, really, with the tides, major tidal effects. And so we have to consider that as a factor, because as you probably know, bull sharks are able to go into fresh water, and so that's not as much of a factor for them. And so just to get into how this works, we use long-term tracking with stationary acoustic receivers and coded acoustic tags. And so the image on the top right shows just the range of sizes. This is from a company called Vimco, of their tags. I use the two largest tags, the V13 and the V16, for bonnetheads and bull sharks respectively. And then the receiver, which I showed you a photo of earlier, is essentially just this black cylinder with a little node on the end. That's the actual antenna. And then we had two anchor designs. One was just a fixed cement block and a PVC pipe, and that was moored to the bottom essentially, and had a buoy up to the surface, so we could just go by boat and pick it up, pull it up on the boat, and unclip it and download the data and perform maintenance. And then we had a fixed monitoring receiver in this bullet float or buoy that was anchored to the bottom. This was no buoy going to the surface here. We had to dive on these to retrieve them. And this was because of shrimp trawling. So we had to design a receiver anchor that could be trawled over. So the idea is that these basically just fold down as a trawl goes over them, and they pop back up. So the tags were surgically implanted. It sounds kind of rough, but it's actually really easy for a shark. It's a small incision, 1 to 2 sutures tops. We make that laterally or on the ventral side. And then the tag battery life was 1.5 to 2.5 years. You have a lot of control here when you design your tags from Vimco. The tags can last up to 10 years. However, you have to compromise battery life and power. And because of the high flow, high turbidity, meaning low water clarity, tons of suspended particles in Apalachicola Bay, and that this was part of my graduate studies, so there is sort of a timeline on this, we optimized for shorter life tags with high power to maximize the probability that we would detect the animals. And so in that picture on the left, this was my student Ashley Doughty performing tying the sutures. Early on when we were tagging sharks, I would do the incision and pop in the tag, but my clumsy hands full of adrenaline, like we talked about stress response, I turned it over to her. She had more dexterity with her smaller hands and was better at tying the sutures, so I let her do that. So you can see us doing surgery there, and then we've got an undergraduate volunteer just lightly pouring water over the shark. I prefer to use a bucket instead of a hose, because a lot of people love to like shove a hose in a shark's mouth, but if you don't temper that water flow, you can actually damage their gills by just blasting water over their their mouth and gills. So we opt for just a little bit of flushing with a bucket of water to keep them aerating and keep them cool. And so this is our receiver array. Every yellow dot was a receiver in Apalachicola Bay. You can see that they're spread pretty wide. This was a collaborative project with a lot of goals. So this was not specifically designed by me for this project. So I kind of had to make do with what we had, but that's what the array looked like. And if I don't have any questions about this part, I wanted to give a chance just in case if there are any questions on the methods and sampling, but otherwise I'll move on to some of the results. I should say, if you're
tell me again,
curious,
those yellow dots are where you had all the acoustic receivers?
That's where the receivers are. Yep.
Right.
Yeah.
How did you go about deciding where to put them?
That was probably a year of going back and forth with other scientists, optimizing habitat coverage. So we tried to prioritize monitoring, soft bottom habitats, seagrass habitats, and then oyster reefs or hard bottom. We also tried to cover the inlets. This was the array at the end of the study. There's about I want to say a dozen receivers that did not survive and were either stolen or more likely missing. Unfortunately, our study, when we deployed the array, this was before Hurricane Michael, and Michael did a number on us, unfortunately. So, for instance, if you can see my cursor in West Pass, we actually had 2 receivers down here that were yanked out of the bottom, never to be found. And so essentially prioritizing habitat coverage, trying to cover the inlets and outlets to monitor immigration and emigration in and out of the bay for a lot of fishes. And then, essentially just trying to compromise with other scientists as you do to meet everyone's goals and try to design something that'll work for more than one species.
Right.
Yeah, and so
how many tags did you get deployed?
I will get to that.
Okay, great. I'm cutting ahead.
Yeah, I'll get to that. There were, I think, 32 receivers in the end, and yeah, I'll get to tags. One thing I should have mentioned is alongside this work, we had 2 other companion projects. We had, I was doing fishery independent surveys, so essentially doing longline and gillnet surveys for sharks, which provided a lot of our tagging opportunities. And then I mentioned my student Ashley was doing an undergraduate honors thesis that she just got published a few weeks ago, actively tracking bonnethead and bull sharks. And that's when we actually lived on the boat. We tracked a shark for 52 hours and followed it around with a similar but slightly different tagging technology. And those provided a lot of tagging opportunities as well. So for some of these animals, we actually double tagged them with active and passive acoustic tags, followed them around for 2 days, and then had at least 1 or 2 years of data after that as well.
Very nice.
And so I got to give a shout out to FWC and their fishery independent monitoring program. They provided the blue crab data. They have monitored Apalachicola Bay for many years with trawl and 2 types of haul seine surveys. And so rather than reinventing the wheel, I borrowed their data graciously. And so just a shout out to Dave Gandy and the FWC FIM program. And so they provided the blue crab data. I converted that to catch per unit effort. So that's a simple standardization we use in fishing data, where we either standardize it by the time the gear is in the water or the spatial coverage. So here it's fish per 100 meters squared of trawling coverage. And then the size ranges were chosen to reflect the selectivity of the sharks. And so this is a figure from Enrique Cortez's paper from 1996, where he looked at, and this is specifically blue crabs, shark precaudal length, which just means the length to the base of the tail, and then crab carapace length. And you can see there's kind of a middle ground there where the smaller and kind of medium-sized crabs are more popular for the bonnetheads. And so to get to Lance's question, I was able to tag 20 bonnetheads and 20 bull sharks. That actually took 3 years, 2018, 2019, and a little bit of work in 2020. We were lucky to be able to continue our work throughout the pandemic, thanks to the wonderful staff at the FSU Marine Lab. We only detected, however, 12 bonnetheads and 18 of the bull sharks. You can see the size range here. So we tagged some pretty small, these are in centimeters, small bonnethead sharks. So probably, 1.5 foot to 2.5 feet long or so. Our sex distribution was heavily weighted towards females. We catch a lot more females than males out here for some reason. And we had as few detections as 78 for one individual and as many as over 3,000. The bull sharks were a little more productive in terms of the data. Their size range was right at 1 meter to 180 centimeters. So probably coming up on 5.5, almost 6 feet long for the largest. Sex distribution was almost even at 9 males and 11 females. We had a similar minimum detection number of 74, but for some of our bull sharks, we detected them actually over 28,000 times. Now the tags are pinging every 20 seconds or so. So those thousands of detections, if they're kind of patrolling around a receiver in that habitat and hanging out for hours, and they're going to really rack up those detections. And this is just another photo, kind of a weird perspective of me with one of the last bull sharks that we tagged. And so I have a couple maps here showing some individual sharks, and I did this just because I wanted to show you kind of what we would expect from one individual. You know, obviously the goal here is to study a group of individuals and make some broad conclusions about what they may do as a species or a population. But just to give you an idea of like, hey, we tagged, you know, STIB number 1, what did it look like? How does it compare to some of the others? And so this is tag number 3323, which is a 74 centimeter, about 3, what would that be? 2.5 feet, 2 feet. Bonnethead, and she was detected over 3,000 times in 2018 and 2019, mainly on just 4 receivers. And she was also actively tracked, so that red X marks the location she was tagged. And so you can see that for this bonnethead, there were a lot of detections, but really only on that one set of receivers. And then we had bull sharks that kind of showed a similar pattern where we had a lot of detections on just a few receivers like this, or we had this large male that was a 200 centimeter male that was detected on almost every receiver in the entire system, almost 5,000 times. And yeah, so you can just kind of see the idea of the difference here. So one of the things that we like to look at when we use this acoustic data, we kind of answer some other alternative questions. And one of the things that I was hoping in this study is that sharks would be coming back year after year, so that if we tagged them in 2018, they'd come back in 2019 and we get a whole nother year of data from them. And for the most part, that was true. We had site fidelity in both the bonnetheads and the bull sharks. And so this is just a plot looking at, you've got the months and the year on the x-axis on the bottom, and then the sharks and their IDs on the y-axis. And the dots are just when they were present. And so you can see that you've got these kind of like pulses here in the summertime when the bull sharks are showing up. And what's really interesting is for some of the juveniles, the winter between 2019 and 2020 was rather mild. And so it appears that they may have actually been resident year round. And so they may only migrate or emigrate out of the bay when temperatures get too low. And if they don't get too low, they may not have to. And so that's the first thrust here of our basic results, how, kind of the goals of the study, our methods, and our main research questions. And so I'm going to start talking about the main analysis now. I'll talk about 2 analyses. One are network analyses, which is a way we just look at how each species used the array, which receivers they moved to and from, and in what order, which receivers were the most popular. And then I'll move on to some statistical modeling, where we actually tried to quantify some of this stuff and get at our main question of, was there an effect of predator and prey distribution and density on the bonnetheads? So the first thing I'll talk to are network analyses, where we have one network for each species. So we pooled all the detections for each individual. We had to bin the detection, so we used only the summer season from May to August, and then detections were binned in 6 hours just to kind of condense the data, because you get so much data from these tags. And then we constructed an adjacency matrix for movement between receivers or presence at the same receiver. And so an adjacency matrix is basically taking pairing every receiver with every other receiver and looking at movements from receiver A to receiver B and how many times that happened. And that creates a whole matrix of data that we use in the network. Then each receiver is represented by what we call a node. It's just a circle on the plot, and paths among receivers are represented by weighted edges, which is a fancy way of saying a line drawn from each node. And the thickness of the line represents how much that path was used. And so I should preface this, network analyses did not originate in ecology, and these are being adapted. And so you can see some of these kind of, the terms we use aren't really intuitive, especially for a biological question, but that's the background there. And then we calculate a couple metrics to describe the networks. The metrics are the nodes, the degree, the total number of connections. Edge density is the proportion of all possible connections or edges in use. So that means of all the different combinations of paths, how many were actually used by those species, and then the average path length. And so here's our first plot of a network. This is for the bonnethead sharks. And so just to orient you, the axis of the array is kind of tilted. The network seems to just have to do that. Looped arrows mean that in a 6-hour time bin, in the next 6 hours, that shark was still there. Otherwise, you can see some of the paths that they took. The larger the circle means the more common that node was used. And I did color code the nodes. So the green nodes represent seagrass habitat, which the bonnetheads tend to really like. The brown nodes represent mud. And you can't see, all of our inlets are represented by a blue color that you can't really see. So the edge density was 0.04, which means the bonnetheads were using a very little of the possible space in the array. They're really only kind of honing in on these couple areas and then making these little sort of foraging trips to the other sites. And so one thing I'd like to point out is there's a big sandbar at these two popular receivers here. And then every receiver that I've circled these are receivers targeting seagrass habitat. So as I mentioned, they picked up a lot on these receivers because they're in there foraging on seagrass, or not on seagrass, foraging on blue crabs in the seagrass. That's a whole nother can of worms, where some folks think that bonnetheads are herbivorous and grazing on seagrass, and I have my own opinions about that, and we can talk about that later. Then for the bull shark network, you can see their edge density was 0.16, so they used way more of the whole space in the array. You can probably just see that in just the nature of the plot, where you don't have one or two really giant circles and a bunch of teeny tiny ones. It's more even, and so they're going all over the place, and they're using a lot of the possible paths between receivers. And some of this is intuitive because bull sharks are a larger animal, so we can expect that they move more just in terms of body length and everything, but this was still really interesting. And so just to remind you to compare the bull shark and the bonnethead networks.
So can I ask this? Can we just go back for 1 second? Just to make super clear, I want to understand everything really well. So we look at the map down here on the bottom, right? And each of those yellow dots is an acoustic receiver. So then if we take that to the plot of data, so each one of those circles, the bigger the circle, the more frequent that they were there. Is that right?
Correct.
Okay, but each circle, and the ones that are just simply black dots, they maybe they didn't go to. So it kind of is associated along the same axis here. So
Yeah, it's just
the red dot on the far left side over here represents the same, if you look on the map, at the
correct.
very
Yeah,
tip on the
that
left side
is.
over there, right? Okay.
Yeah, Lance, that's I'm not. I tried so hard to get these plots to use the latitude and long data just to twist it, and
Yeah.
I really struggled with it, and so I eventually just went with it. So yes,
I get it. I get it.
they're,
It's a good
they're all
spatial
spatially arranged.
representation. Yeah.
It's just twisted up a tad, but yeah, you're right. The red circle on the farthest left when you're looking at the plot is the westernmost receiver in the map.
Right. Okay.
Yeah.
That makes sense. That makes sense. And so the thicker the line, the more commonly that shark was there, right? Loops represent them
Right.
staying there, and straight lines
Yeah,
represent them going from one point to another.
exactly.
That makes sense.
Exactly. And
Yeah.
so those thicker lines basically mean that that's a common path. Now, there's some nuances to this. What it really means is we detected, that's the order in receivers chronologically they were detected on. And
Okay.
so technically, you know, I'm sure bull shark movements aren't all these straight lines from all our perfectly arranged points, right? It's just showing you, you know, the order that they showed up in. So we don't know what they're doing between one or the other, but it still gives us a really good idea of what they're doing. And what I like that you mentioned is seeing those loops when you know they're staying in the same places. And so that was especially true of bonnetheads, like this giant kind of fat loop coming off this main receiver. That's in Pilot's Cove. It's this beautiful seagrass bed. It's quiet. It's protected from much of the rest of the bay. There's a ton of stuff in there. And the bonnetheads really seem to like that. That was true of any of the bonnetheads that were on the western side of the bay. They really liked that habitat.
Cool.
Okay, so another little breather here, and I'll get into some of our statistics and modeling. So I've included everything so you can see it. I'm not going to talk about it all in detail, but I'll try to explain how we did it, what we put into the statistical equation, and what we took away from it. And so one of the answers to the sub question before of are blue crabs heterogeneous or homogeneously distributed, meaning are they evenly distributed or are they patchy? And they were definitely patchy or heterogeneous. So this is just a plot of the FIM data, and you can see just Florida's FWC's Fishery Independent Monitoring Program there, led by Dave Gandy in Apalachicola. And so we can see very clearly that there's these, you know, blue crabs are heavily distributed up in the northern reaches of the bay. They occur throughout the bay. Obviously, they're along the coast and everything, but you can see that it's not perfectly even. And so to look at this, we took a subset of our data where the bonnetheads occurred on the western side of the bay to kind of control in that space. So we took a subset of 6 receivers, and we used an analysis called linear mixed models. And so we chose these receivers because they were present throughout the study period. So they were never removed by the hurricane. They never had to be replaced. And we also chose receivers that were separated fairly well from one another, or at least representing different habitats. So we didn't have any spatial autocorrelation, which means that when, you know, say for instance, this patch of 3 receivers, we could have used all 3. But when a shark's in there, they're more likely to be detected on the other receivers just by the nature of being there at the first. So we didn't want to kind of weight the statistics too much. And so to quantify bonnethead habitat use, we used the proportion of individual detection days. They had to be detected a minimum of 6 detection days. And then blue crab density was calculated using all sets, FWC sampling sets within 1.5 kilometers of each receiver. That was designed to essentially look at the detection range of the receiver. So one thing I didn't mention is that we do range tests on all of this. So when we deploy the gear, we manually set out tags just in little mesh bags at regimented distances, sequential distances away from the receiver to look at our detection range and our detection probability, so we have an idea what we're kind of starting with, if you will. And then bull shark presence probability was the proportion of all bull shark detections at those receivers. And so let's see, this is just showing you the names of some of our receivers, so I'll walk through this just a bit. This is in Apalachicola Bay proper, and so we had All Tides Cove, Sykes Cut, and Pilot's Cove. Then we had Dry Bar. This is called Indian Pass or St. Vincent Sound, East Bay, St. George Island Bridge, and Cat Point. And so these are some of our results here from the model that I'm not going to talk about too much, but blue crab density and bull shark presence were correlated. And so in this plot in the lower left, I have blue crab density on the x-axis and the probability of bull shark or sea-loo presence on the y-axis. And you can see that bull sharks were more likely to be present in the areas that blue crabs were the most dense, and that makes sense because blue crab density is really high up here in this freshwater area, or this, sorry, not freshwater area, but river-dominated area where you have more freshwater influence, and so I think bull sharks were more common up there. We used maximum likelihood estimation in the model, and the variables were standardized to a mean of 0 and variance of 1. This was just to simplify the statistics. And so I have each site here in this table and some of the numbers for the density of the crabs, the presence probability of the bull sharks, and then mean bottom salinity, which was really important because salinity was lowest at those northern receivers in East Bay, where the density of blue crabs was highest and the probability of bull sharks present, was also highest. So that's an important factor to keep in mind. We included a random effect in the model for individuals. So we had 6 bonnet heads, and we want to try to incorporate the idea that there is individuality among those individuals, so or among those sharks. So this is in the model that's just going to throw some noise in the statistical model, so that we don't assume each individual is the same, because technically they aren't, right? And then there was a term for spatial autocorrelation included in the model. We evaluated model with AIC and normality assessed via diagnostic plot. So this was kind of the industry standard, the Ecology information criterion. Kind of in the woods here on the statistics, but now you know how this all goes. So the really interesting thing here was that bonnet heads were not detected at the highest density prey sites. That was over here in Indian Pass or St. Vincent, then at the power line in East Bay and Cat Island. They were obviously detected at the places I mentioned at Pilot's Cove, All Tides Cove, and at Dry Bar. And so this is just our champion bonnethead shark. So there is also a significant negative coefficient of bull shark presence probability on proportional bonnethead habitat use. And this is a slide out of my dissertation defense. So that's a really wordy way of saying that there was a significant effect. When you go through grad school and you've got a committee that's going to try to ping you, try to phrase stuff like really specifically. And so that's why this is, I tried to really be explicit at what we looked at. But the thrust here is that there was a significant effect of bull shark presence on where these bonnet heads were being detected. And I did include salinity in the model. Salinity did not have a significant statistical effect, but I wanted to mention that because that's like to me, the most obvious question about this is, what about salinity? And it looks like salinity wasn't a major factor. And in fact, since we've done this study, we've learned more and more that bonnet heads actually can tolerate much lower salinities than we once thought. And so we saw salinities coming down, down to, this is in parts per thousand or but salinity down to basically the 20s. It can be as high as 30 to 35. So St. Vincent Refuge was essentially full strength salinity. And then up in East Bay at the power lines, it was about 20, but that's well within what bonnetheads can tolerate. And so we think this is a real pattern. We don't think this is influenced too much by environmental covariates or that kind of thing. The low salinity could be a stress factor. So in addition to the presence of predators, other things, it could be that this is just a really stressful environment to be a bonnethead in because salinity is low, water clarity is super low, and there are predators around. And so just to make some conclusions, the relative importance of prey density in driving habitat use may vary. And I'm sorry, that's from a different study. Blue crab density does not predict bonnethead habitat use. Bonnethead habitat use may have a negative relationship with bull shark presence probability, and the interaction increased bull shark presence in areas of lower salinities. And so what that means is I'm sorry, that's what I missed. There was a significant interaction of bull shark presence and salinity, meaning that they were probably correlated. So I didn't actually quantify that, but the idea here is that in the areas with lower salinity, you had a relatively higher chance of encountering a bull shark. And so that was that interaction there I'm referring to. And this results supported by the foraging theory according to ideal free distribution, which I didn't talk about under predation risk, but the idea that you are not going to be going in perfectly distributed, the sharks are not going to be distributed in a perfect way, and they're not going to necessarily be going into the optimal habitat when under the risk of predation. And that got a little wordy at the end. I'm sorry about that, but just some acknowledgments for the project. And basically, just for this one, I acknowledge my advisor, Dean Grubbs, was extremely supportive, as well as the staff at the FSU Coastal and Marine Laboratory. I imagine this audience is mostly located in South Florida, but if you're ever up here, the FSU Marine Lab is not very well known, I don't think, in the greater scheme of things, but it's an amazing place. It's a really great group of people. They do open houses every other year. It's an awesome place to visit. And just want to acknowledge FWC, Dave Gandy, and Bob Gorecki for sharing the blue crab data. And then, of course, NOAA and especially Save Our Seas Foundation, who are the primary funders for the project. I did get support from Florida Sea Grant, as well as the Guy Harvey Ocean Foundation after the Save Our Seas support. And just have some follow-up slides in case you have any questions, but that's it for me.
Awesome. Well, it's really amazing. I know that for a lot of our audience, some of the scientific jargon, I guess you could say, can get a little dense, but I think that's good though to present this sort of data and to really know you've done your due diligence in studying this, right? So when you know what you're talking about when you say it, because you've done a lot of the analysis, which is really impressive, right? A lot of presentations kind of gloss over that part, but I think I appreciate you including it. So thank you for that. We're gonna turn off the slide there for a second so we can see each other. Yep. So in the end, bonnethead sharks were affected by the predation risk of bull sharks, right? Mm-hmm. And so I think that's a pretty amazing, I think, piece of information that you were able to glean from that, right? I mean, to be able to look into this species and be able to tell some that detailed information is quite impressive, I think. So kudos to you for getting that done.
Thanks.
Yeah. We had a few questions from the audience. We had one question about temperature. They were asking about the temperature of the bay in general, but do you think temperature could have had any sort of relationship to the sharks? Does the water at different temperature coming out of the river as opposed to what it is in the bay itself?
Yeah, that's a great question because generally temperature is a major influential factor for shark movement and habitat use. There actually was not, and one of the reasons we constrain the study to the summertime. Even cutting out some of the spring was to control for temperature. One of the neat things about Apalachicola is there's so much water flow that the bay actually doesn't get as hot as St. George Sound, the area to the east, and temperature stays pretty homogeneous. So there can be some really crazy haloclines where you have low salinity water at the surface and high salinity water at the bottom, just because of the density of salt and everything and how it separates. But temperature is pretty homogeneous, so we don't think, and I did include temperature in some of the early models, and they weren't an influential factor, a useful variable. So that's a great question, but don't think it's a factor here in the summertime. Yeah.
Nice. And then we had another question about, you showed that distribution of when they were there, and you said that a number of the individuals you were able to see year after year, which was really nice. Glad you didn't waste your tags.
Yeah. Me too.
But where did they go when they're not there? And
I love that question.
How long of a period are they typically gone for?
Gone. Yeah, excellent question. And so for some we can't answer, right? But this is one of the really cool things about these studies. Something that folks can kind of be aware of is there are a ton of acoustic receiver arrays, and we're part of the ITAG network, which is another thing that's run by FWC. So shout out to them and the FWC Research Institute. Where we have a collaborative network. So if your animals are detected on somebody else's receiver, they will let you know. And so most of our sharks have been detected in South Florida. They've been detected in Tampa Bay. They've been detected offshore of Tampa. Some of the larger sharks have been detected way offshore and like hundreds of meters of water.
These are the bull sharks.
These are the bull sharks, yeah. Right. Same thing with the bonnetheads. We would expect them to be going south, but along the coast, so not just going straight through the Gulf of Mexico. They make some, Brian Keller, who's another Grubbs Lab graduate and well supported by Save Our Seas, did a bonnethead migration project, and they on the East Coast, and they made these major coastwide migrations. So I did not have any bonnethead detections in other arrays. So I'm not sure how far they're going or exactly where. Unfortunately, we had fewer bonnetheads be detected and fewer bonnetheads coming back, and obviously they're more risk, right? So they can be eaten. They can also be harvested, be lost to bycatch and that kind of thing. And so it's tougher to say for them, but we've had other species that also go south, like lemon sharks, black tips do this. And so usually if somebody's doing it, other species are following suit.
That's interesting. Yeah. That's a really interesting collaborative effort. I didn't realize the state had such a wide network of acoustic tag stuff in the water. That's really neat. And it's so nice that they share their data with you, right?
It's really great. There's also the FACT array on the Atlantic on the East Coast. So they have websites if or the folks that attended. Yeah, it's FACT, FACT, and ITAG, just ITAG. But those are, they have, I believe they have some interactive website kind of stuff, but just really neat networks.
We'll have to look it up and share it with the audience. ITAG FWC, I see it now. Okay. I'll drop the link in the chat. So we have a few more minutes for a few last minute questions, about 5 more minutes or so. And I know that your journey has kind of started with you growing up in Tennessee. I'm partial to that. I grew up in Tennessee as well, starting with frogs and salamanders, just like you. Migrated down to Florida to study sharks. You and I share a very similar journey, except that you went on and did your PhD. I went and taught in the classroom instead. But so in case there's anyone interested in following a career in fisheries, in science in general, could you kind of give us a synopsis of why you got involved in this, why it still piques your interest today?
Sure.
Yeah, your general journey.
Yeah. Well, as you mentioned in my introduction, I kind of just got interested in fishes naturally, just being, I was always interested in life and critters and that kind of thing. And then I decided that, I kind of followed the the idea of pursuing marine biology as an undergraduate, and then I got really interested in the idea of just doing science that served a broader purpose than information for the sake of information, which is important and worthwhile. But I really got interested in management, and then my path to doing research actually started. I got a position as an undergrad working with oyster larvae in a hydrodynamics lab, and I was in a position where I could volunteer my time where I could. If I had spare time, I'd try to get out with other grad students to get experience. I was able to snag that job. I made myself as available as I possibly could, which isn't always easy, if you're working and supporting yourself in school and that kind of thing. But trying to get involved as an undergrad just to get experience. And then I would say my first kind of big break was an internship at Moat, and that really set me on the path of sharks and that kind of thing. And then it was just, getting the grades and trying to do research and build some experience to go to graduate school. And, I think there's a lot of ways to get there. I think you have a lot of freedom in undergrad that you really don't have to major in marine biology. It can be biology, it could be not be biology. But the biggest thing is trying to get experience. And my advice to anyone that's like considering this field or is a little bit younger is just to try to do as many different things as you can. And see what you like. And sometimes that's hard to do with how much time you have and everything, but the field is moving more, I think, away from unpaid internships as much as it can to paid opportunities, or trying to increase the representation and diversity in marine science. And that's a big issue, but a lot of times, funding's limited. We need a lot of hands. So I was absolutely dependent on undergraduate volunteers as a grad student. But yeah, that was my path is, and then the other thing I would say is, like I said, oyster larvae. I've had students that told me like killer whales are nothing. Like, I'm really sorry, but that's a top tier species that only very few people ever get to work on. And so you've got to be open-minded and recognize that when you're early in your career, research is research, and hopefully it's good quality and all, but don't turn it down because you think it might be boring or you're not interested in whatever it is. You need that experience, and you can specialize later. So that's important.
That's a good piece of advice. Yeah, and it's been a common answer across all of these series from other guests as well, to kind of ask questions and just follow what you're interested in, and just go after an opportunity. If there's a volunteer opportunity to go help a researcher on a boat, go.
Yeah.
Right. Yeah. Well, that's cool. That's good. Thank you for that. So for the rest of the audience that maybe isn't maybe a little further into their careers and maybe more vested in their careers and can't just switch and become a fisheries biologist, what can we do to help advance conservation, especially of marine coastal habitats? We have a very important coastal marine habitat here in South Florida. We have Biscayne Bay National Park, and Florida Bay on the other side. What would be some ways we could get involved in helping to conserve some of these places and some of these really important species?
Yeah. So I think, there are obviously like little practices like reducing your use of plastic and things like that we all know about. The other things I would really advise are engagement with coastal conservation groups. Coastal cleanups are really important. And then we're at I think a crossroads in Florida where we have many interests politically, and I don't mean to turn this in a political direction, but having some awareness of what's happening, how at a local, county, or state level that policies are being implemented that affect these things. That's really where we're at. And so just being engaged and trying to understand the risk. Coming to talks like this is a great way to be educated, interacting with the museum, and then just kind of spreading the word to other people and trying to, I grew up with an interest in this stuff, and it took me a while to realize that not everyone is inherently interested in this or has opportunities to go to the beach or whatever. And so one of my favorite things to do as a graduate student was work in our the Saturday at Sea program, which works with school kids just to show, like, take them out trawling and show them the habitat and what's out there. And and tell them about it. And so from a fishery perspective, you hear a lot about sharks, their threats worldwide, but domestically, we do great. And a lot of people may not want to hear this or realize this, but our shark fisheries are very well managed. We have sustainable fisheries for pretty much everything that is fished or harvested and protected. And so at home, our threats to these animals are mainly through habitat destruction and the destruction of crucial juvenile habitats. Seagrasses, mangroves are extremely important for things like lemon sharks or the endangered small-tooth sawfish. Depend on mangroves, which are kind of always under attack for coastal development and these kind of things. And so that would be my advice, that at any career point or stage of life, that you could try to get involved in there, and then vote with your mind and your environment.
Yeah, that's good advice. I really appreciate that, and just being more aware, right, of what's happening around you, and
the scale
yourself.
that affects these processes. It took me a long time as an adult to realize that, like, I got to pay attention at a lower level than national news and mainstream stuff, just because of who's making the decisions that affect this kind of thing.
Right. Yeah. All right. Well, thank you so very much for your time this evening. We really appreciate you coming on and highlighting your research. It's really impressive what you've been able to do, and congratulations again on your PhD completing it.
Thank you very much.
It's a very
I appreciate being invited. This was a lot of
endeavor.
fun.
Excellent. Yeah, we're happy to have you back anytime. So thank you all for joining this evening. We'll call it there. For next month on August 4th, we have another distinguished speaker series from the Save Our Seas Foundation. We're going to be talking to researcher Jeff Whitty from the Sawfish Conservation Society. So Cheston just mentioned the importance of mangroves and sawfish. Next month, we're talking about them. Providing Sawfish Education for Global Conservation in Action is the name of Jeff Whitty's project and his presentation. So please join us then, 6 to 7-ish on August 4th for that presentation. And in the meantime, please, if you're local or not, please feel free to come on into the museum, learn more about all of our natural ecosystems we have here in the museum. All right. Thank you so much. Again, Dr. Peterson, thank you very much for joining today. And
you're quite welcome. Thank you, everyone, for coming.
Yes, thank you. Thank you. We look forward to next time. Thank you so very much.
Thanks, y'all.